start once again. I'll record this video for children who could not reach out. So today we are discussing about cell organelles of the cell, and the first cell organelle is endoplasmic reticulum. The name endoplasmic reticulum, discovery of endoplasmic reticulum is done by Keith Porter. And the cell organelle, which is starting from nucleus and it is extending into tube-like structures, is called as endoplasmic reticulum. If you see, these are continuous sheet-like structures. They are not, see, we are cutting the nucleus like this and seeing it looks like this. But this is a sheet that extends, okay? Continuous sheet-like structure, okay? Suppose if you take one reticulum, this I have drawn there. So it is a sheet like this, okay? Sheet like this. When I am drawing another branch for this, like this is one sheet, already it is there. So this is second sheet, like this, it is continuous. When drawing 2D diagram, it cannot be understood, but these are sheets like this. Among those sheets, sometime what happens? These sheets will extend into tube-like structures like this. These tube-like structures that arise from these sheets are called as tubules. They are all tubules. Okay? Structurally, if you see, there are sheet-like structures called cisternae, tube-like structures called tubules, and sometimes they form small pouches like this. These are called as vesicles. Okay? The cisternae, tubules and vesicles are structurally components of endoplasmic reticulum. If you see this endoplasmic reticulum called cisternae has small ribosomes studded on it like this. You cannot see these studded ribosomes on vesicles or tubules. Only on cisternae it is found. So this part is called as rough endoplasmic reticulum. Rough endoplasmic reticulum. This part, these two come under smooth endoplasmic reticulum. Smooth endoplasmic reticulum. Okay, so this rough endoplasmic reticulum has ribosomes. Since ribosomes are present, it helps in protein synthesis. Protein synthesis. This smooth endoplasmic reticulum, which did not have ribosomes, helps in lipid synthesis. Lipid molecules are produced. So that is about structural features of this smooth and rough, different endoplasmic reticulums, components of endoplasmic reticulum, and coming to functions of endoplasmic reticulum. Since it is forming, it is spreading into the cytoplasm. It gives some mechanical strength to cell, mechanical support to cell. In the cytoplasm, there is nothing, only plasma membrane is there, nothing is there inside the cell. The cell needs some support to stand, bro. That support is provided by some other structures also. This also, endoplasmic reticulum also gives that strength to stand for a cell, right? It helps, it spreads inside the cytoplasm and helps in conducting substances. Conducting substances in cytoplasm from one location to another location, from nucleus to outside, like that. It helps in protein synthesis. Smooth endoplasmic reticulum helps in lipid synthesis. Rough endoplasmic reticulum helps in protein synthesis. Not only synthesis, it helps in storage also. Those substances which are produced in endoplasmic reticulum are stored for some time. Storage of substances. Storage of synthesized substances also occurs in the endoplasmic reticulum. Right? Very simple, nothing complex about it. Very simple structures, very simple to understand also. Next, we'll go to Golgi complex. So, Golgi complex. Golgi complex is the name. Golgi complex is given by scientist Camillo Golgi. His name is Golgi only. Okay, Camillo Golgi, and he discovered Golgi complex and named it. This is also called as Golgi apparatus. Golgi apparatus. In case of plants, these structures are very simple and they are called as dictyosomes. They are called as dictyosomes in plants because they are very simple in the plants. Okay. Now coming to the structure of Golgi complex, at the end there are small vacuoles like this and extended by this cistern. Okay. These are the vacuoles, cistern. Vacuoles, cistern. The newly formed ones do not have that vacuoles, simply cistern. When it is growing, when it is completely developed, these vacuoles are formed. Sorry, vesicles. I am confusing you. Sorry. This is vesicles are formed. Vesicles, these are cisternae. Even this is also cisternae only. This end ones which are bulged or vesicles. Sometimes when the work that is to be done by Golgi complex is completed, then they separate from themselves. Okay, they get separated from this Golgi complex. This is called as vacuoles. Okay, what is dictyosomes? In plants, Golgi complex is simple and it is called as dictyosomes. Dictyosomes are simple Golgi complex. Golgi complex. Present in plants. Okay. So that is the structure of this Golgi complex. So this side it is called as cis. This side it is called as trans. Trans side old ones are there, old cisternae are there, which have fully developed vesicles. Sometimes these vesicles separate and they form trans structure. So what is the function of this Golgi complex? Function of this Golgi complex, if you see, first, it helps in lipid synthesis along with smooth endoplasmic reticulum. Second, major function is packing of proteins. Packing of proteins. Proteins will get packed. So, after the proteins are synthesized inside the endoplasmic reticulum, through small vacuole-like structures, they are passing into Golgi complex. Inside the Golgi complex, these proteins are packed. From there, it will be carried further. So, here it is useful for packing. And in sperm, what is sperm? Do you know? Male gamete. Male gamete has one head region. Above that head region, there is a very sharp pointed end called as acrosome. This acrosome helps the sperm to penetrate into ovum. So this should go and poke into the ovum like this. So that poking function is done by the presence of this acrosome here. That acrosome is synthesized by this Golgi complex only. Okay, in plant cell, when the cell division occurs, there should be first, before formation of cell wall, One structure is formed between two cells. This is called as middle lamellae. Middle lamellae will form first. After that, primary cell wall will form. After that, secondary cell wall will form. Okay. So this middle lamella is also produced by Golgi complex. Middle lamella is produced by Golgi complex. So these are the functions of Golgi complex. Lipids, it helps in lipid synthesis along with smooth endoplasmic reticulum. It helps in packing of protein. Protein should be packed well. If they have to function well, they should be packed well. That function is done by this Golgi complex, and then it also helps in acrosome synthesis, middle lamellar synthesis. So those are the major functions of Golgi complex. Coming to lysosomes, the third organelle we discussed today is lysosomes. Lysosomes are very simple round structures like this. There is no complexity in this, very simple round structures. And inside this, what we have? Enzymes that digest. What they digest? They digest carbohydrates, proteins. They digest carbohydrates, proteins, nucleic acids, fats. Nucleic acids means DNA and RNA. These all substances are digested by the help of some enzymes. Those enzymes are present inside this round structure called lysosomes. Okay? So, sometimes when the cell should die, there are different reasons why cell should die. When the cell becomes old, it will die. If the cell identify itself to be a default, or it is generating some mutant, means something which is causing cancer, then it kills itself. That is called as autolysis. Okay, autolysis of cell. Okay, programmed cell death. Programmed cell death. So when one cell is producing another new cell during this cell division, the cell monitors very closely. There are certain proteins inside the cell which help them to monitor closely if there is any default in it or not. The moment it finds some default or some cancerous growth, immediately the cell will prepare itself for programmed cell death. This process is called as apoptosis. Apoptosis. During these reasons, when the cell becomes old, when the cell should go autolysis because of some infection, some default like that, or during the synthesis of new cell there is some fault, then it undergoes programmed cell death called apoptosis. At that time, what this lysosome does is it releases all the enzymes into the cell. The cell is made up of mostly carbohydrates, lipids, and proteins. All membranes only, no. Endoplasmic reticulum is made up of membrane only. Any membrane has the same organization by lipid molecule, by layer of phospholipid molecule. Proteins are embedded, certain carbohydrate side chains are there. So you take the cell membrane, you take the membrane of endoplasmic reticulum, nuclear membrane. Other organelles, we'll study now, few more organelles. All those organelles except ribosomes are having envelope around it. Ribosomes and centrioles don't have any envelope around it. All other organelles have envelope which is made up of phospholipid bilayer, proteins and certain carbohydrates. So once these contents are released outside, carbohydrates side chains will digest, proteins will digest, nucleic acids which is present in the center of cell will digest, fat molecules will digest. It lead to the death of entire cell. So these lysosomes are also called as suicidal bags. Suicidal bags of cell, suicidal bags of the cell. Whenever the cell decides to do commit suicide, means self-killing. Whenever it want to kill itself because of any reason, the enzymes are released outside and it leads to the complete complete damage of the entire cell. Okay. So the lysosomes can be it is there are stages in the formation of lysosomes. When it is not completely formed, it is called as primary lysosome. It separates itself from the Golgi complex. First of all, it separates itself from the Golgi complex. So when it is able to form proteins, these enzymes are nothing but proteins. At that time, it is called as secondary lysosome. Or after the contents are completely formed, at that time it is called as tertiary lysosome. And when it is releasing all the contents, it is called as residual lysosome also. Residual lysosome. So depending upon the stage in which it is there, it is called as Kaushik. Anything to say? Kaushik Rama. Anything to say? So when it is in the initial stages, it separates itself from this Golgi complex. At that time, it is called as primary lysosomes. When the proteins start building inside, it is called secondary lysosome. When the complete protein formation takes place, it is called as tertiary lysosomes. When it releases its contents, it's called as a residual lysosome. So that is about lysosome, children. So So still we have some more organelles to discuss. We'll end it here. We have discussed the three organelles, endoplasmic reticulum, Golgi complex and lysosomes are completed.